The central dogma of biology is the mechanism where parts of the DNA is transcribed into mRNA strands that are then translated into amino acid chains, which fold into protein. However, the genes transcribed from the DNA do not always share the same expression. Constitutive genes are necessary for daily cell functions and are expressed or transcribed into mRNA at a constant rate. Conditional genes are not required unless the cellular environment or developmental stage changes, and are therefore only expressed at certain times. So, how do researchers study gene expression? Many methods exist in molecular biology to analyze gene expression. Such techniques include microarrays, reverse transcription PCR, qRT-PCR, northern blots, and reporter genes that I will describe in the rest of this video. So what is a reporter gene? Reporter genes monitor the levels of expression by fusion with a promoter or a gene of interest. It's not a dye. Reporters are genes that encode a protein indicator that allow researchers to localize in which cells or tissues the gene of interest is actively expressed, where proteins are transported or localized, and the relative stability of the mRNA strand or protein. The first type of reporter genes are transcriptional reporters. Transcriptional reporters indicate tissues where the promoter of a gene of interest is active. In the example shown above, the green fluorescent protein reporter gene is fused only with the promoter region, and should transcription and translation occur, the green fluorescent protein fluoresces within those cells. The second type of reporter genes are translational reporters. Translational reporters are fusions of native genes of interest and a reporter gene that encodes a chimeric RNA transcript. The chimeric mRNA is then translated into a single amino acid chain. The amino acid chain then folds into a single protein containing two parts, the native protein of interest and the reporter. This fusion protein allows researchers to determine subcellular localization, transport, and the relative stability of the native protein through an observable reporter. So which type of reporter genes are commonly used in research? Many types of reporters exist to study gene expression across different species of organisms. Commonly used reporter genes use color changes to monitor the level of gene expression. These include fluorescent, luminescent, and chromogenic reporter genes. The most common reporter genes are green fluorescent proteins and their derivatives. Green fluorescent protein was first isolated from Aquaria victoria jellyfish, and red fluorescent proteins were first isolated from Discosoma anemones. Currently, many derivatives have been isolated from sea creatures to use in research. Luminescent reporter genes encode the enzyme luciferase, which catalyzes the oxidation of luciferin to produce a bioluminescent reaction. First isolated from fireflies of the Lampyridae family, 3 derivatives are currently used in research: firefly, Ranilla, and Nanoluke. The last type of reporter genes I will briefly discuss in this video are Gus and LAC-Z, which are chromogenic reporter genes. Gus encodes the enzyme beta-glucuronidase to convert X-gluc to a blue precipitate. Similarly, LAC-Z encodes the enzyme beta-galactosidase to convert X-gal to a blue precipitate. This change from colorless to blue visualizes in which cells or tissue Gus or LAC-Z is expressed.